As many of you know, I have spent most of my career studying cytotoxic T lymphocytes and how they control HIV, and now I'm here at a pharmacology seminar talking about monoclonal antibodies. So obviously I've figured out what works and what doesn't work, and if nothing, I'm at least pragmatic. So let me tell you a bit about broadly neutralizing monoclonal antibodies. So monoclonal antibodies were discovered in 1975 by Milstein and Kohler, and that led to them getting the Nobel Prize in 1982. But it was really around 2008 that the technology became available for a real explosion in monoclonal antibodies, and that was our ability to sort individual B cells that express immunoglobulin on their cell surface that we can detect with certain probes. So you can use an HIV envelope or an altered envelope and pull out B cells that express an immunoglobulin that specifically targets that HIV envelope. And then you can amplify that individual immunoglobulin gene and you can clone it and you can produce monoclonal antibodies. You used to have to make hybrids with mouse cells, et cetera, and it would take forever. Just at the Vaccine Research Center, we have developed monoclonal antibodies against RSV, HIV, Ebola, influenza, malaria, MERS, and there are many companies around the world now that are just pouring out monoclonal antibodies against multiple infectious diseases. If you look at the pipeline of available monoclonals that are going into clinical testing, it's clear that this is the new frontier in infectious disease therapy. So what I'll do is I'll give you an overview of existing HIV monoclonal antibodies, then talk about antibody engineering for better potency and breadth, a little bit about Fc mutations to affect antibody PK, and then the lessons that we've learned from protection and treatment trials. So this is a schematic of the HIV envelope, with this being the viral membrane down here. And there are actually 5, but mostly 4 different sites that broadly neutralizing monoclonal antibodies can attack. There's the CD4 binding site or CD4 super site. There's a V1, V2 site up at the top. There's a V3 super site. And then there's the membrane proximal external region or the MPER region that's down near the membrane. There is this interface site, but it's less well characterized. This is a list of all the different monoclonal antibodies that have been published, and this is actually not a complete list, but it shows you that there are multiple antibodies targeting these different sites, and the ones that are circled here are the ones that have actually gone into production and are being, have either been into clinical trials or are being prepared for clinical trials. So you can see that those 4 major sites I talked about have monoclonal antibodies that are being developed and are being set up for clinical trials. This is a slide that shows both potency and breadth of monoclonal antibodies to the 4 different sites: CD4 binding site, the V3 glycan, the V1, V2, and the MPER. So this isn't all of them, but these are some of the major ones. The way this is shown is you have 208 different viruses that each antibody has been tested against. And the IC80, so the concentration of that antibody that will inhibit 80% the entry of that virus into cells, is then plotted on the y-axis for these 208 different viruses. The number up here is the percent of those that are completely resistant. So you get no neutralization at all. So what you have are some antibodies that are very potent, and the potency is shown here. The bar is the mean or median IC80 against all 208 antibody or viruses, and then this number up here is the percent that are not neutralized. So that's really the breadth. So here you have the potency, and here you have the breadth, and you can actually plot this in a different way. So you can take that breadth and plot it on the y-axis versus the potency on the x-axis, and what I show in these 4 antibodies. These are old antibodies which were discovered in the old method before we had lots of new antibodies. And what you can see is that they're not very potent, and some of them are not very broad. But these are what we used to think were the broad and potent, broadly neutralizing antibodies. And then these are the new antibodies that we're dealing with now. And what you can see is that they're all much more potent, and some of them are extremely broad. But in general, as you get out to these that are the most potent, they tend to lose breadth. And ultimately what we want is this gold star out here. Something that is as potent as any of our antibodies and as broad as any of our antibodies. So that's the target that we're going to shoot for. So how can we actually take these monoclonal antibodies and make them more potent and give them better breadth? So let me go back to this slide and show you VRC07 523. This is actually an antibody which started out as a cousin of this one over here, VRC01, which had 13% resistant viruses. And through antibody engineering, it was made to be more broad, so only 4% are now resistant. And you see that the mean IC50 is about 5 or 10 times more potent. So just by changing amino acids through the crystal structure, studying the crystal structure of this antibody, we got something that was more potent and broad. And the same thing over here with an MPER antibody. So this was the parental antibody, which was very broad, but not too potent. We increased the potency about a tenfold. So if we go back here to the potency breadth curve, here's VRC07, which I told you was very similar to VRC01. Here's VRC07 523. So we've increased the potency and breadth. Here is the original 10E8, and here are the new variants of 10E8. So once again, much more broad and potent, but still we're not over here where we want to be. Another way that we can improve potency and breadth is just to combine antibodies. So this shows 4 different antibodies, and this is another way of showing potency and breadth. It's similar to the other way, but every time you get neutralization against a virus at a different concentration, you just tick up this curve. And so where you don't get up to 1 here, that's the fraction that's not neutralized. And the further to the left, the more potent you are. And so you take these 4 colored lines, and those are the 4 individual antibodies. Then you start combining any 2, you get these brown lines, which all shift to the left and go more to the top. Any 3 antibodies are the gray lines, and all 4 together gives you the black line, so that you can get greater potency and breadth just by combining antibodies. Now you can either do that by taking and giving multiple antibodies, or you can use technology to actually put more than one specificity onto a single antibody. So this is the cross-mAb technology, which was used by David Ho's group, where you actually put one specificity on one arm and another specificity on another arm. What he did is he took anti-envelope, so a neutralizing antibody specificity on this arm, and then an antibody against either CCR5 or CD4 on the other arm. And this just shows the data. So this is the mAb, the anti-CD4 antibody. Here's the Pro-140, which is an anti-CCR5. And then you have, with one arm being an envelope specific, and then adding either the Pro-140 or the mAb on the other arm. What you really need to look at is over here. If you take 10E8 and you add the other specificities onto the other arm, you get really incredible increase in potency and breadth. The other way that you can do this is you can actually put 3 specificities onto an antibody. And this is work out of Sanofi. So one arm here is VRC01. On the other arm, they take 2 variable regions and put them onto the same arm. So in this case, 10E8, an MPER, or PGDM 1400, which is a V1-V2 antibody. Now, it looks as though you would say, oh, well, this will be accessible. This out here will be accessible, but this specificity, that's not going to be very accessible. But actually, if you look at the crystal structure, so here's GP120 binding to VRC01. That's very accessible. These other two are actually opposed in space and both very accessible. So here's PGDM1400 and its binding site over here, and 10E8 and its binding site over there. So actually all three sites become very accessible. And when you look at these potency and breadth plots, what you see is that these are three different antibodies that were made in this fashion, and because of manufacturability concerns, this is the one that was actually chosen to go forward clinically. And if you look at the potency breadth curves on these, so you have the three individual antibodies, VRC01, PGDM1400, or 10E8, in these curves, and then the trispecific is here. So once again, increased potency and breadth by putting all three specificities onto one antibody molecule. And then if we go back to this plot and we say, how do these new trispecific antibodies look? Actually, we're now getting out towards what we're hoping to achieve. Very good potency and very good breadth. Okay. What about the pharmacology of these? Most antibodies human antibodies have a half-life of about 15 days. But what is known is that the Fc region of the antibody is involved in recycling of that antibody, and specifically the region that binds the neonatal Fc receptor, which is this region in here. And you can make mutations in the region of the Fc that binds the neonatal Fc receptor. What that does is allows that antibody to be taken up into endosomes, but remain bound to the neonatal Fc receptor at low pH. So that antibody then gets recycled and released back into the circulation. And it increases the half-life immensely. So there are a few of these different types of mutations that can be made. The one that most people are making right now is called the LS mutation, because you change these two amino acids to an L and an S. And so we did this with our monoclonal antibody VRC01. In black, you see its PK, a half-life of about 15 days at 5 milligrams per kilogram sub-Q or IV, or 20 milligrams per kilogram IV, or 40 milligrams per kilogram IV. And then in the colors, you see the VRC01 LS. And what you can see is that with this LS mutation, at 5 milligrams per kilogram, either sub-Q or IV, you can maintain over 10 micrograms per ml for 6 months. And then at 20 milligrams per kilogram IV, you can maintain over 50 micrograms per ml, and 40 milligrams per kilogram over 100 micrograms per ml. So now you have every 6-month dosing of these monoclonal antibodies, where you can maintain levels that will hopefully be therapeutic, and it'll all depend upon the sensitivity of the virus that you're trying to attack. So in the last 10 minutes or so, let me tell you what we've learned, at least what I think I've learned, from protection and treatment trials. So let me start with protection trials. There's a long history of giving monoclonal antibodies to non-human primates and then challenging them with SHIV and showing that we can protect. But actually there's virtually no human data regarding passive protection of HIV monoclonal antibodies. Because of this, we have recently gotten involved in the passive antibody prevention trial. This is a phase 2B efficacy trial. It's called AMP, for antibody-mediated protection. And it's designed to address the issue, can passively infused monoclonal antibody prevent HIV-1 infection in high-risk adults? And this is conducted as a joint effort between the HVTN and the HPTN. VRC01 is the antibody that's given. It's a CD4 binding site antibody. It's given at 30 milligrams per kilogram or 10 milligrams per kilogram IV or placebo, and it's given by IV infusion every 8 weeks. There are 2 trials. One is in high-risk men in North and South America, and one is in high-risk women in South and East Africa. Ultimately, the goal is to enroll 4,600 subjects. As of about 2 months ago, we were up to 3,725. So we're up to about 80–90% enrollment at this point. So by summer, we will be fully enrolled in this trial. The trial is designed to ask what serum level of monoclonal antibody is associated with protection, and it's powered to define an overall 60% efficacy. So what happens if this is successful? Well, if we show that we can actually protect with passive infusion of a monoclonal antibody, we will be able to define the level of plasma monoclonal antibody that's needed to protect against infection. Is it 5 micrograms per ml? Is it 10, et cetera? And that we can then translate that into whether we can give subcutaneous administration of monoclonal antibodies rather than IV. This would really improve self-administration. Can we go out now to 6 months, every 6 months with our LS antibodies? If this works, then there's a big incentive for the pharmaceutical industry to start developing the next generation of monoclonal antibodies. And we can also look at other ways of delivering these for long acting, so by DNA, by AAV vector, by RNA. And knowledge that neutralizing monoclonal antibodies actually protect will help the vaccine field. We now have a target. We know that if we get this level of neutralization in serum, we'll protect. Well, let's shoot for that with our vaccines. But the other thing that will be important is that this will start to help us develop a target product profile. And based upon what we think the results of the AMP trial will be, we've actually started to develop a target product profile for monoclonal antibody prevention. We feel that we'll probably need, or it would be optimal to have 2 immunoglobulin, or either 2 mAbs or a single mAb with 2 different specificities. The indication would be to prevent HIV infection against 98% of strains, depending upon whether we're talking adolescents or adults or infants of HIV-positive mothers. We have different targets there. We want to give subcutaneous every 3 to 6 months so that we can allow self-administration. We want very low adverse frequency, and we want it to cost less than $50 per person per year. And these are targets we think we can get to. But it really hinges on the human efficacy data and commercial interest. And the reason I bring up commercial interest is that pharma partnership is going to be very important. So development of a preventive monoclonal antibody would be greatly facilitated by big pharma interest. Big pharma is obviously mostly interested if there's a therapeutic indication, but a therapeutic product could then be used as a preventive agent. If you look at the Truvada PrEP scenario, developed as a therapeutic, academics then took it and said it works as a preventive, and now it's marketed and approved also as a PrEP agent. But the target product profile for a therapeutic agent will be different from that of a preventive, and it will likely require coverage of viruses by at least 2 monoclonal antibodies to avoid escape. There are multiple different ways that you could use monoclonal antibodies in treatment. You could use them during acute therapy to try and knock down the reservoir. I'll go to the bottom one. You could use it to try and reduce the cell-associated reservoir, and our next speaker will be talking mostly about that. But I think the big area that I see as the hottest area for use of broadly neutralizing monoclonal antibodies is to maintain long-term viral suppression. after that's been induced by antiretrovirals. So once you knock down virus replication and you're well suppressed, can you maintain that suppression with every 3 to 6 months injections? So let me just go through very quickly a number of different clinical trials, phase 1 clinical trials that have been done with broadly neutralizing antibodies, and the results are all very consistent, as you'll see. So here's the first one that was published. This is 3BNC117. This is a CD4 binding site antibody. When it was given to people who were viremic, the virus came down. After about 2 weeks, it came back up. Those who had lower virus load, the virus stayed down longer. Here's VRC01, the same format. What you see is you give the monoclonal antibody, the virus goes down. After about 2 weeks, it comes back up. In those who had lower virus loads, the virus stayed down longer. Here are 2 individuals where we didn't see inhibition. If you look at those 2 individuals and you pull virus out of all of these individuals, those 2 individuals who did not suppress, they had virus that was resistant to VRC01 to start with. And so they didn't suppress very well. Here's a different specificity. Those 2 were CD4 binding site. This is a V3 glycan antibody. You give the antibody, the virus goes down for about 2 weeks, comes back up. Goes down for about 2 weeks, comes back up. If the virus load was lower, it stays down longer. Here are 2 individuals who didn't respond. They had resistant virus. So all very consistent. A couple of other trial scenarios have been done, and that is to take people who are suppressed. And then around the time that you take them off antiretrovirals, you give broadly neutralizing antibodies and you ask, can the broadly neutralizing antibody maintain suppression? So here's the first trial with 3BNC117, again a CD4 binding site antibody. The antibodies given as antiretrovirals are stopped. They maintain suppression, and then the virus comes up as the antibody's coming down. What you can see is that sometimes the virus comes up when you still have quite a bit of antibody, and sometimes the virus stays down and only comes up after the antibody is quite low. But like in this case, clearly, even with lots of antibody around, that virus just came right up. So what ends up happening when you look at this, here are individuals who weren't given broadly neutralizing antibody in terms of the weeks to virus rebound after ATI. And here's those that were given 3BN117, and you can see there's a delay in rebound. The same thing was done with VRC01, and here's the time to rebound in these individuals. Here's the antibody concentrations when they rebounded, and as you can see in most of them there was still quite a bit of antibody around. And basically in the 3BNC117 trial and here in the VRC01 trial where you're giving a single antibody, the answer is this. That over time you will develop resistance, and resistance to a single agent will limit the ability of these single monoclonal antibodies to maintain suppression. So what we've seen is that there's been rapid advancement of potent HIV monoclonal antibodies into clinical trials since their first major discovery for these broadly neutralizing ones in 2009. They're classically used to prevent infection, and we have a big phase 2 prevention trial ongoing. Multiple phase 1 treatment studies have been completed with single monoclonal antibodies, but it's unlikely that single monoclonal antibodies will be able to maintain long-term viral suppression because of resistance. Combinations engineered for potency and breadth or multi-specific antibodies in combination may get over that issue. In addition, combining these with antiretrovirals will be helpful, and there are trials being planned to combine VRC01 with cabotegravir. But I think regimens in which virtually every virus is covered by 2 antibodies, with or without a long-acting antiretroviral in addition, will probably be needed. And I'm just going to end on this slide, which I think depicts the problem. So what I show here are the 208 viruses are on this axis. Here's one antibody, PGT121. Here's another one, PGDM1400. So if you have an orange or a yellow or a red, that virus is neutralized by that antibody in these different concentrations. If it's blue, it's resistant. And so what you see is these 2 antibodies have completely non-overlapping resistance patterns. So these viruses are resistant to this antibody but not this one. These viruses are sensitive to this antibody but resistant to that. So you combine them, you say, hey, this looks great. Look, now we're catching all of these viruses. But in fact, you're only covering all of these viruses with a single antibody. And if we're going to get to a point where we have coverage with 2 antibodies of virtually every virus, then we're going to need a combination like this. And these are 2 very broadly neutralizing antibodies, a CD4 binding site antibody and a MPER antibody. And I think that really needs to be the basis for our combination therapy. And then we can add one of these others in there so that we get triple coverage of some. But at least the vast majority will already be covered by 2 antibodies. So with that, I'd just like to thank all these people who were involved in helping to develop these antibodies and helping me put this talk together. And I'd be happy to take any questions. Thank you.